All right, Krishna, you're good to go.
Okay. Hello, welcome everybody. Uh, on behalf of the Southern California section of the American Chemical Society, I extend a hearty welcome to you, audience, and to the speaker, Dr. Katherine Bay. And before I give a brief introduction about Katherine and Schrödinger, I have to convey my thanks to the local section activities committee of American Chemical Society for offering an IPG, that is Innovative Projects Grant, for us. And today's speaker is Dr. Katherine Bay. She is a senior scientist responsible for leading the Teaching with the Schrödinger program. She develops academic curricula that integrates Schrödinger's molecular modeling tools in the classroom. Prior to joining Schrödinger, she was an assistant professor of organic chemistry and earned her PhD in organic chemistry from UCLA. And a word about Schrödinger. Schrödinger deals with molecular discovery, better therapeutics, and material science. And in addition, they offer online certification courses which may involve practical learning and hands-on application of workflows using Schrödinger technology. They also offer free learning resources, step-by-step instructions for performing tasks and analyzing data. And they also offer teaching aids, teaching software in the classroom to learn theoretical and applied science. And the company is located in New York with 10 office locations. Today's topic on which Katherine is going to talk about is science at Schrödinger. Over to Katherine.
Thank you so much, Krishna. Hi, everyone. My name is Kat, and I am a senior scientist from how Krishna described it, a computational chemistry and biology software company called Schrödinger. And I'd like to thank both Krishna and Danny from the Southern California section of the American Chemical Society for organizing this seminar and also for inviting me to present. Before I get into my presentation, I just wanted to share a little bit about myself to show that scientists are really multifaceted and can have interests outside of working. I am a dog mom to a rambunctious 7-year-old pug named Bugsy. And in terms of hobbies, I really love to travel and bake and try new foods. I'm also a Southern Californian native. I grew up in Arcadia, California, and went to Arcadia High School. I then got my bachelor's degree in chemistry from the University of Laverne. And while I was an undergraduate student, I was really lucky enough to do several different research internships at schools like Caltech, and I even traveled abroad and did chemistry research in the UK at the University of Cambridge and also in Japan at Nagoya University. And all of my research experiences collectively really solidified that I wanted to pursue a PhD in chemistry. So I applied to a lot of different universities for graduate school and ended up going to UCLA to get my PhD in organic chemistry. So if you have any questions about applying to colleges, graduate programs, or even how to apply for a research internship, I'll save some time at the end of my presentation. So feel free to ask me questions about any of those topics. And then after I went to grad school, I became a professor at my alma mater at Laverne and taught a variety of different undergraduate chemistry courses. And then I landed at Schrödinger on the education team. So Schrödinger is a company made up of people who have studied computers, science, and human health, and we really draw on experts of various fields so we can all work together to solve a wide range of different problems. I work with many different kinds of scientists like chemists, biologists, immunologists, educators, and even students. And one of my favorite things about working here is that I get to work with folks who have so many different skill sets and who bring different perspectives all towards working towards a common goal of improving human health and the quality of life. So I know that all sounds great, but what do we actually do to make this happen? So at the crux of it all, everything around us in our environments, all the things that we use, our phones, TVs, planes, cars, the food that we eat, even a medicine or a drug that you take is made up of molecules. And these are the same atoms that make up the periodic table of elements. And today I'm going to discuss how we at Schrödinger can use computers to design new drugs in a systematic fashion. So if I were to ask you about how many molecules do you think are possible to even make? For context, there are about 10 to the 46th power of molecules of water in all of Earth's oceans. And that sounds like a lot of molecules, right? So I know this question sounds nearly impossible because how can you even know how many molecules even exist in the world? So I'm going to make this a little bit more specific. So how many drug-like molecules do you think it's possible to make? And you're welcome to put your answer in the chat. And remember that when you're thinking about what does drug-like even mean, we want the molecule to be made up of atoms that consist of things we do not want in our body. So no molecules that have um, elements like arsenic or any heavy metals because those are toxic in the body. So we want to narrow the number of possible molecules actually available to a number that is actually drug-like. Are there any guesses? I'll give you a clue. It's something to the power of 10. Any guesses? 10 to the 10, 10 to the 80th, 10 to the 100th power. Any guesses? 10 to the 20.
100.
Yeah, 10 to the 20 is a good guess. 10 to the 100, also great guesses. Answer is there are about 10 to the 60th possible drug-like molecules. Basically, this is another way of saying there are way too many possibilities to actually try when we're trying to think about how many drug molecules are actually possible, which is mind-blowing even by Jimmy Fallon. So all great guesses. Thank you for participating. So another way to think about this is the image on the left-hand side is called a principal component analysis plot, or a PCA plot, and this shows about 19 million of those possible 10 to the 60 drug-like molecules. So every single colorful dot that you see in this image equals a distinct molecule. And the further apart any two dots or points of molecules are in this image, that means they're more chemically distinct or different. So a situation that's very common in research is that, say you're trying to solve a problem by finding a molecule that you need for your research project. And let's say the dot that you're looking for is all the way in the pink area, but your starting point is somewhere in the green area. So really, in order to get from the green area to the pink area, one way to approach this is you could walk yourself from green across the yellow region all the way to the pink area in order to investigate. But that would take a really long time because that's a lot of molecules you have to investigate to end up at the point that you want to get to. Another way to approach this is you can jump around and try random molecules, almost like buying a lottery ticket. You'd have to be super lucky to land in the spot that you want to be in, or even be even remotely close to the spot that you need in the pink region. Or another approach that you could try is, what if you could try all of the molecules, or at least a majority of them, to see if the drug molecule you're looking for is in that swath of chemical space? And that's kind of what Schrödinger software in a nutshell aims to do. It's to give researchers the ability to evaluate a large amount of chemical space and use different techniques to triage yourself into the area of chemical space that you want to be in. And oftentimes there's a balance of computational speed and scientific accuracy. And unfortunately, that's usually an inverse balance. So the faster you get a result, usually the less scientific rigorous is that calculation. But that doesn't mean it's not useful. Because if I want to evaluate 19 million or a billion or maybe multiple billions of molecules and possibilities, I don't want to spend an hour. I probably don't even want to spend a minute or even a second going through every single molecule. But if I can quickly triage myself into the right space by balancing different techniques at different times, then we have a real chance of finding the molecule we need in a time frame that we need it. So Schrödinger at its core is a physics-based computational company. And we do integrate aspects of machine learning. And in fact, you'll see it in a bit throughout my talk. And these two scientific cores are quite complementary, but that's something that I think is going right back to the underlying physics that underpins the natural phenomena that we see all around us. So really that sets up the scene. There are a lot of possibilities that we can work with, and we need to allow scientists to be very efficient with how they use their time because what they're looking for is extremely specific, especially in the realm of drug discovery. And really what we're looking for is one molecule to solve all of your problems. So when we think about this, a situation that researchers often find themselves in is maybe we have an initial starting point of a molecule and it doesn't initially check all of the boxes that we need it to check. It's really just a starting point. And so we make a modification to the molecule and we try to get more of those boxes to be checked. We're really trying to get an optimal property space here, and you can go on and try all of these different things, and maybe you're going in the right direction, but one small tweak in your molecule could set you back many steps. So a lot of this doesn't work sometimes. It's really hard. For an example, let's say if I were to say, all right, you come to me and say, hey, Kat, I have a headache. Could you make me a medicine? And I'll say, yeah, sure thing. No problem. We got this. Here's a medicine that I've created for you. It will make your headache go away, but it's going to make your eyebrows fall off. And you're going to say, you know, I'm good. I'm going to stick with my headache because I want to keep my eyebrows. But these are all properties that we have to think about because you want to create a drug molecule that does only the one thing and nothing else. And that's really hard. So then I come back to you and I say, okay, I've tweaked the medicine. Here's a new medicine for you. You get to keep your eyebrows and your headache will go away, but it's a shot. You have to inject the medicine every single day. And you're probably like, I'm really bad with shots. I'm going to pass on that. And that's another property to think about when you're designing medicine. How is the drug going to be delivered? When you're thinking about designing new medicines, it's not just finding the molecule that's going to do the thing that we want it to do, but it's also thinking about, will this medicine have any deal breaker side effects? You probably have seen commercials on TV about new medicines by pharmaceutical companies where they tell you what the medicine is. And at the very end, in the last 5 seconds of the commercial, they say the long list of different side effects that this medicine could cause. So that's not ideal. Also, when you design new medicines, you want to think about, will this interact with any other potential drugs that a patient might be taking? Is it easy to take? Like I said, pill versus an injection. Those are things that you want to think about because if patients don't want shots, there's no point in creating this fantastic drug molecule if the only way to administer it is via injection. Another property to think about when you're designing new medicines is how long is the drug going to stay in your body? Do you want to take this headache medicine every hour? Not really, that's not ideal. But at the same time, you want to take the medicine and have it be excreted in a timely manner. You don't want that drug to stay in your body for months, also not not great. Another thing to think about is, do I want to take this medicine and have it metabolized into something that's potentially toxic? That's not ideal as well. So there are so many different properties that we as scientists need to consider, and that's all really what makes designing medicine extremely difficult. And when we look at this in terms of a cost standpoint, this is a lot of money to optimize a molecule to get all of those boxes checked. And that's probably the most expensive part of the whole drug discovery process. So Schrödinger's technology focuses on what we call the preclinical part. So it's all of the first four rows of this table here. So everything that comes up to getting a compound or a molecule to get into clinical trials. And we all just lived through a time where everybody was very invested in some clinical trials and the speed at which a certain vaccine was able to progress through. So this is really where we're focusing on here. And again, we've heard a lot about how costly and time consuming and difficult this is. So unfortunately, that headache medicine that you want to design, you're also going to need about $2.5 billion. Oh, and on top of that, you're probably going to have to wait 10 to 15 years on average. So I hope you're okay with that, because on average, it takes about 12 years to design a new medicine. Luckily, this is shifting, but it really is what it comes down to. We want to change these things, and designing new medicines is too much money, and it takes way too much time to get medicine to people who need them yesterday. And there's always a lot of other complications that come into the picture as well, because at its core, science, as I'm sure you all have experienced yourself, is not just working with molecules, but it's also working with humans. And we as humans make decisions in really weird ways. We lose information, we forget things, we have biases that we also need to mitigate. And so there are a lot of other ways that the scientific process can get disrupted as well. But there is some good news. So if we look at how many medicines have been developed over time versus the cost of that, you can see in this graph that we've been steadily losing bang for our buck. And somewhere around the year 2000, we kind of crossed over into making less than one medicine or drug per $1 billion. However, around the 2010s, that started flattening out, which is what we want. And another way to think about this is about our mission at Schrödinger. We want to turn this around. So it's great that it's flattening out, but we actually want to reverse this trend. We want to get medicines or molecules to people faster, which also means cheaper and better outcomes. Again, this is very aspirational, and you're probably wondering, what does that mean? How does this technology even work? Because if you Google Schrödinger, you probably will come across a story about a cat, which I will get to in a bit. So just for a little bit of scientific history, starting off with Isaac Newton, famous for the apple falling on his head. Isaac developed many theories to explain the world and space. And with Newtonian physics, space and time were absolute, and you could figure out everything about them. Next comes along Albert Einstein with his discoveries of relativity, and everyone realizes then that space and time can actually be altered by matter, usually when things are either really small, like subatomic particles, or when things are really big, like space. Then comes along Erwin Schrödinger. He shows up with quantum theory and says, guys, it's even worse than we thought. It's so hard to figure out anything absolute. And this is Schrödinger's equation, which describes the movement and location of an electron. And this is really tricky because an electron can behave like a particle or a wave. And Schrödinger discovered that it's the way that you observe an electron, and that actually affects how you observe it. In other words, the way you run your experiment can influence the results. So knowing where electrons are and how they will move is really important when it comes to being able to simulate molecules on a computer. When you draw a molecule on a computer, you need to be able to have your one drawing represent all possible shapes, properties, and interactions that the molecule can have, just as it would be like if that molecule were in a test tube. And you're probably thinking, even Neil deGrasse Tyson thinks that this is mind-blowing. But just to preface, no actual cats were harmed. That really was just a thought experiment to illustrate Erwin Schrödinger's somewhat crazy idea that an electron can be a particle and a wave at the same time. So the theory was a cat is inside of a closed box and you can't see it. Inside the box, there's a flask of poison that could break if you open the box, and that would kill the cat. So as long as you leave the box closed, the cat is effectively both alive and dead at the same time. And Erwin Schrödinger used this thought experiment to illustrate how something can be two things at once, and how observing the cat or opening the box would fundamentally change the nature of the cat you're trying to observe, very similar to an electron. But we really haven't fixed any problems here. Drugs take a lot of time and a lot of money. And at the same time, physics is super hard. So what are we going to do? Luckily, Schrödinger gave us a lot of different tools. And with Schrödinger's equation, we can extrapolate out and try and get a sense of where electrons are in space, which gives us a sense of how molecules might behave and how they might interact, what their properties might be, etc. And all of that is really useful information despite being very complex. And it's hard just for a single electron, but imagine the things that we're worried about are much, much bigger than a single electron system. So in the end, there's still a lot of math. We have a lot of physics that we have to contend with. And when we start thinking about different atoms, especially atoms collected together, it starts to get really complicated. And so we need to think about what are ways that we can improve that efficiency. So computers are really good at calculating complex mathematical equations, but even this is something that's a bit hard to do. So we need to think about how we can balance this very scientifically rigorous, rigor, and also the costly calculation with maybe some cheaper versions of this, where we can substitute in some estimations for really rigorously calculating the exact same thing. And like I said, we're not just interested in a single electron or maybe even a small molecule, or perhaps something that could be a bit larger, like a drug molecule or medicine, or even a whole protein or biologic. When we consider larger systems like this, we're going to have to start weighing up our options here. So predicting how this will work is complicated, but the good news is that there is something that we could do by balancing that computational speed and scientific accuracy. So this, let me make sure it can play here. Don't know if you can hear that, but that's okay too. Can you hear? Hopefully not. It's okay. The audio is not as important as long as you can hear.
We can hear.
Oh, you can?
Yeah.
Let me just lower that volume. So this is a cartoon representation to give you an idea of something that we might want to do with the software. So say this protein is involved in some sort of disease, and we want to modulate how this protein is working. Maybe it's working too much, maybe it's not working at all. So we're going to get a small molecule to try and fix this protein's function, and we're going to kind of scan through it quickly to just see what molecule might fit here, almost like that lock and key analogy that you've probably heard of in your biology or chemistry classes. Now, once we find a good starting point and start triaging that large swath of chemical space, then we can really start to do more scientifically rigorous calculations to get us more into the energetics, maybe get us more information about those properties that we need to balance for this molecule here. And once we're able to find it, then we can hone in and fine tune more computationally rigorous details to get a really strong sense of what's a good drug candidate for which all of those different boxes that we want to check when we're designing a new medicine. And there are a lot of different ways to do this. So we might want to start with figuring out, is this the right protein target? Is this the lock that we really want to pick? then we need to figure out, okay, we have a lot of keys. What are the properties of these keys? What's the key that's going to work, and what will fit in the lock that we have? Will it have the right properties that we're looking for? And only then will we be able to think of it moving forward into something like clinical trials. Other things to think about are, how do I want people to use this key? Meaning, is this going to be a shot or a pill? Is this going to be a big pill, hopefully a small pill? What's the ideal dosage? And would it have to be taken hourly or daily? So there's a lot of different properties to think about. And all of these different tools that are offered by Schrödinger software can help mitigate those. With all of these different tools, they all help scientists get the information that they need to make good decisions to move their research forward. And we have a lot of different ways that we can do that. So within Schrödinger, we have our own internal drug discovery team. And this is really fantastic because they kind of keep us honest, and we say, okay, we can develop a lot of tools, and we also can develop software that does fun things like create these nice images and movies and whatnot. But you're probably wondering, does this actually work? And we're really excited to say that so far it has been proven so. So our internal drug discovery group, they're working on a project where they said, okay, we have a target for a disease, we've validated it, now we need to try and find that key molecule. We need to evaluate that large swath of chemical space and try and find that one molecule that's going to get us to solve the problems that we're trying to solve. So they created what's called a ligand library, which is just consisting of a lot of different molecules from that chemical space that they want to test out. And their library consisted of 8.2 billion compounds or molecules. And here's where we get a mixture of physics-based and machine learning technology. So some of these molecules, actually, the vast majority of these molecules were designed by machine. So with that said, this is kind of what we want when we're trying to build a library. So you can think about, that's more than the population of the world. So then we have to find that one person in the whole world who's going to be able to do what we want it to do. So here's where we start balancing more physics-based calculations and triaging that down to a workable number. Now we're going to get to even more rigorous with our calculations, but more expensive to get it down to roughly a street of people. And here's where we start bringing in wet chemistry and working with other groups of scientists to actually make these molecules in a lab and make sure that our predictions computationally are on point. And then, happy to say that experimentally, they were able to find that one molecule. They found that one person out of the whole world that was able to do what they needed to do. And this whole process of idea to candidate usually takes somewhere between 4 and 6 years. But with the use of computational tools with Schrödinger software, our team was able to do this in about 10 months. So that's super exciting, especially thinking about that $2.5 billion and the 10 to 15 years that we're trying to get to reduce it by. So now this molecule has moved into the clinic, and you're able to do a Google search of this inhibitor that we designed, and it's now in phase 1 clinical trials, all started from the computer. And it's really exciting that we're able to get it this far. It's not a done deal yet. It's still in the process. Nothing really is approved until the FDA approves it, but this is all still very promising. And most importantly, it's happening with far less time, far less money, and far less waste when you're going into this type of research. So you're probably thinking, that's really cool, but how can I learn about computational chemistry? So like Krishna mentioned in the beginning, we offer a variety of different online certification courses that really contextualize the learning of key ideas through real world problems and applications through different curated pathways that we've designed. And all of our courses include both theory and practical use of the software through a variety of different learning tools like videos, step-by-step tutorials, and also exercises that integrate computational tools as well. And all of our online courses are self-paced, so they're asynchronous. They provide temporary access to our web-based version of our software, so you don't have to download anything. So it makes it super convenient to get started, and they're all meant for folks who might not have experience using computational tools before. So we have a variety of different courses that our team has designed. Some are introductory, some are advanced for folks who have never used computational tools before and kind of just want to dabble in this, to just learn more about this exciting growing field. I would recommend any of our introductory courses. If you enjoyed today's topics, I would recommend the Introduction to Molecular Modeling and Drug Discovery if you want to kind of get your hands on and see more of what this looks like through looking at large swaths of chemical space and designing potentially new drugs and inhibitors. Feel free to check that course out. And there's a lot of different ones as well. And I'll provide my email at the end if you want to contact me to learn more information about any of these courses. Here's just a little glimpse of what one of our courses looks like. So all of these figures show different elements of the course. If you're interested in learning more about our online learning platform, we have examples of our videos, our tutorial exercises. The software that we use is called Maestro, and you can see a snippet of what that looks like in the bottom right-hand corner. And if you would like to learn more about computational tools in your classroom, we also have a bunch of different lessons that we've created across various chemistry courses. So we have general chemistry, organic chemistry, and medicinal and biochemistry lessons as well. And all of our lessons have different computational exercises that you can test out. And with that, I would like to thank the entire Schrödinger Education team. We are a global team across various offices throughout the world. I'm based out of the San Diego office, but our headquarters are based out of New York City. We are a very diverse team of scientists, and it's really great working with everyone on a variety of different educational materials to help the next generation of scientists. And with that, I've left a lot of time at the end for questions from folks. Thank you so much for giving me this opportunity to talk. If you have any questions that you'd like to ask me about anything that we talked about today, my email is on this page. You can email me at katherine.bay@schrodinger.com or our whole team at education@schrodinger.com. And with that, I'd be happy to take any questions.
Feel free to ask questions. Probably, Catherine, I will begin with my own question.
Sure.
We know simple organic molecules exhibit stereochemistry for optically active compounds. We have not all optical isomers are not drug candidates. For example, a D isomer may have activity and L isomer will not have activity. Does Schrödinger have a way of actually predicting the stereochemistry and how a particular stereochemical structure would be more active or will give you more response to a particular type of medicine development?
Absolutely, that's a great question. So oftentimes a particular stereoisomer will have a favorable interaction when it's docked into a protein target's active site. And usually a certain stereoisomer will, in the more active conformation, will have, let's say, a more favorable hydrogen bond or some form of intermolecular interaction that is causing that particular pose or bind to be better than the other stereoisomer that probably does not have that particular interaction. So you're able to qualitatively see the differences between, let's say, the R versus S isomer when it's docked inside of that pocket of the protein. And you can almost lay the two isomers on top of each other with the software to see the differences in what those interactions are there or not there, depending on which isomer you're looking at. So oftentimes the activity is dependent on what interactions are forged by that particular isomer or not.
Okay, that's good.
Hi,
Yeah,
Hello.
go ahead. Go ahead.
my name's Sophia. I'm from Pacifica Christian, and I was just wondering, what projects have you yourself worked on, or like, have you worked on certain kinds of medicines? Or like,
Yeah,
what
that's
kind?
a great question. So I'm on the education team. So my project actually is on this slide. So I actually work with a lot of professors, teachers, and students on trying to bridge that gap on teaching them what computational chemistry even is and trying to introduce it more into the classrooms. Because at least when I was a student, I didn't even know about this field until I was in graduate school. So I'm
Oh,
really trying to introduce it more earlier in students' education. So creating lessons in high school and undergraduate school to kind of showcase that this is a very up and coming field and very exciting field to learn about. So I'm not on the scientific research and development side, I'm more on the education side of things.
oh, okay, thank you.
Yeah, of course.
Which components of AI, machine learning or some other component of AI, you think is more useful for fundamental organic chemistry in a classroom?
It's a good question. So for machine learning, we usually use those tools more to develop more drug molecules when we're creating our ligand libraries, just because it takes a lot of manual power if you were to manually change different atoms within a molecule on your own. So we use machine learning oftentimes to speed up that process to, again, cover more of that chemical swath of space. So that's mainly what we use machine learning tools for. And as far as AI, our company hasn't dabbled quite into AI yet, but we are hoping to utilize that in the future as we improve on our software tools. Because we are a software company, we do update our software quarterly. And that's exciting because researchers are always looking for what is the next best and great thing out there. So we want to make sure that we're able to keep up with market demands.
There is a question I've seen from Kim, who is a high school teacher in New Jersey. And she says, I am curious how your software can be utilized to teach chemistry at high school level.
Absolutely. So I've worked with a lot of different high school teachers. They all use it in very different ways. So if you want to integrate it into your curriculum, they use it more to visualize the geometries of molecules, looking at the like VSEPR theory, for example, looking at bond angles, bond length. Taking that a step further, students are able to calculate electrostatic potential maps to see where in a small organic molecule or any molecule really, where electrons are more densely populated. You can also calculate spectroscopic plots. So for example, infrared spectroscopy or IR spectroscopy, looking at vibrational modes of molecules, and you can generate a plot to see where those frequencies are located. So a lot of different ways that can be used at the high school level. Some other teachers, what I've seen, they use it for kind of like capstone projects at the end of their course, giving students a bit more freedom to utilize the software in any particular way that they seem best fit. So within a class, you can group students and assign them a different protein that's associated with a different target disease, and they can kind of design their own drug molecule depending on how their molecule fits into the pocket of that protein target. So lots of different ways that it could be used at the high school level. And I can provide you some more information if you want to send me an email too. My email is katherine.bay@schrodinger.com. Any other questions?
And you have put in there general chemistry also. That means actually from the introductory level, it should be. And do you have some sort of a demo, a demonstration that you can make either in person or through virtual through Zoom to a class
Sure.
of students who are just trying to learn general chemistry and the fundamentals of chemistry?
Absolutely. Yes. So we offer demos all the time for classes. It's one of our exciting parts of our job being on the education team. So if that's something that you're interested in, either myself or someone else from our team can hop on a Zoom call and walk your students step by step on how to use the software, how to draw molecules and visualize them in the 3D fashion, and how to run basic calculations from there. So again, if you are interested in that, feel free to send me an email and we can help you get started with that.
Catherine, you have two students with their hands up. One, the first one was Jessica Liu class, and the next one was Clarita class. They had questions for you.
Cool. Feel free to unmute yourself.
Hi, my name's Casey. Do you see this technology being combined with like protein libraries where the interactions of proteins are known, and like digital thought engines like ChatGPT to create almost like a theoretical experiments, like say you enter like, okay, I want a molecule to do this function, it'll interact with these proteins, and then you put it into like a ChatGPT like program. Do you see that being like a future application of this program?
Yeah, it's a great question. So I'll answer your first one. So what you can do with our software is it's connected to the protein database or data bank, so the PDB. So any protein that is publicly available through the PDB, you can directly import that crystal structure into our software. And from there, kind of like your possibilities are limitless. You can then either dock any sort of ligand library that's either publicly available or one that you designed into that protein directly. We also have tools that look at protein-protein interactions. So far, today's talk has focused on small molecules docking or looking and interacting with proteins. But proteins are super big. And what you also can use the software to look at is two proteins or multiple proteins interacting and what that surface looks like when those two interact with one another. So you can see what those interactions look like. So protein-protein docking is what that's called. So all of that is available within the software suite. And to answer your second question, if any ChatGPT adjacent tools are being looked into at our company, the answer is yes. Nothing is publicly available yet, but that is something that we're looking at to see how these different AI tools can be adapted to the drug discovery space. So not yet, but stay tuned. Clarita, I don't know if your name is Clarita, but feel free to introduce yourself.
I'm Dylan, also from Pacific Education. In the chemical space, correct me if I'm wrong, but there was a blue area, a green, and red. Is there a difference between each area, or is it all applied to each like a chemical space molecule?
So it's all just kind of symbolic. So you can think of like one area of a color being like molecules with rings, or another area of space being molecules that are more flat, that have like sp2 hybridized carbons, or other areas being more aromatic or have more conjugation in their molecules. So it's kind of just subdividing molecules by their properties and also looking at what different functional groups are attached to each of these molecules and kind of categorizing them. So it's not like the pink area is like these type of molecules, it's more just symbolic of how similar they are within their molecular properties. Does that answer your question?
Yeah, thank you.
You're welcome. Alexandros?
Yes, hi.
Did you have a
My name is
question?
Alex, and I'm professor of organic chemistry at the community college here in Los Angeles. It was a great talk, very informational. I hope the students attending learned a lot from this. I've been using molecular modeling for some years in my classes, and I use some software from the company called Wave Function, which is based in Orange County. Just to understand, and I have like 2 layers of the software. Is your software the same thing? It's available? I mean, again, do you have like a student version versus the full version, the research level version? And is that free? Is it open source? Can you please comment on that?
Absolutely. So we do have a free educational version that you can download on our website, but this is more of a visualizer. So you're not able to run any calculations. It's just purely for visualizing. And then we also have
passive side, right? It's not active for a student.
Exactly. And then we also have the Teaching with Schrödinger program, which is the program that I lead. This one you do have to pay. It depends on the class size. If it's a class size less than 60 students, then it costs $45 per student per academic term. And this is a web-based version of our software. It's not a sandbox version, it's the full complete version that students can run any type of calculation with as long as it's for teaching and educational purposes. And that version is web-based, so you don't have to download anything. And then we also have a variety of different research licenses. So if you're interested in using our software for publication purposes, that will depend on what type of tools you're interested in. But you're welcome to send me an email and we can get you more pricing information on that. But we have different tiers of our software that are available.
Thank you.
You're welcome. Any other questions?
There was a question in the chat that I wasn't aware if you had gotten to yet. The first one was from Ara, and it said, can your software be used in general research and product development not related to pharma?
Yes, so a lot of different schools all around the world use Schrödinger tools for their research projects, so that's definitely possible. I would say in terms of software usage, maybe like 40% of our users are in industry and then 60% are in academia. So just to give you a sense of who uses our tools. But it's not just pharmaceutical companies that utilize our software. So I didn't talk about it today, but we also have an entire material science suite. So companies use our software to look at designing different materials that make up planes, TV screens, our phones, looking at different battery materials, because our software is looking at the core of molecules. So anything that is made up of molecules, you're able to simulate with our software. Drug discovery is just one of the application areas, but there's a variety of different other application areas, and you can kind of see that from the different courses that we've designed. So our software can be used for catalysis, organic electronics, polymers, batteries, pharmaceutical formulations, etc. So there's a lot of different applications that our software can be used on. And again, these are courses. So if you want to learn more about how to use it from a beginner standpoint, you're welcome to take a look at that as well. And then I'll answer Nikhil's question. I know that Schrödinger was developed with drug development and organic chemistry in mind, but how suitable is it for inorganic and organometallic chemistry? Well, luckily, just answered that question. It is also suitable for all of those types of materials as well. And we have a lot of different training materials and educational step-by-step videos that also address how you would use this for the first time. From Clarita, what internships did you take prior or during college that set you up on the Schrödinger path? From Maggie. That's a great question. So I didn't really know what I wanted to do as an undergraduate student. I, like many, thought I wanted to go to medical school or pharmacy school and take that kind of health sciences path because that's kind of all I really knew about or was aware about as a student. And it wasn't until I started, I applied to an REU, which stands for a research experience for undergraduate students. And my professor brought it up to me and said, like, you do really well in your lab classes. Maybe you should consider applying to a research internship during the summer. So I thought, why not? I'll try it out. And that's where I kind of fell in love with doing research, and kind of the mystery of not knowing what the outcome was really exciting for me. And I wanted to take that on as I went to graduate school. I realized that I did also an internship at a hospital and realized I'm just like not good with looking at patients, helping patients out from like a direct patient care standpoint. I appreciate folks who are, but that just wasn't the path for me. So I really liked doing research projects. So I took that path. And that's what made me get introduced to Schrödinger software. So I used it in my graduate studies at UCLA, along with other different types of computational softwares. And I thought it was really cool to be able to visualize molecules on the computer and not have to use test tubes and glassware to create these molecules in person. So I thought it was nice that if you make a mistake on the computer, it's not going to explode and you won't get hurt, which is really nice. So I liked that kind of safety net of experimenting, but through the computer. So I was introduced through Schrödinger software there, and I was a professor for some time after I graduated, and I also really enjoyed teaching and interacting with students. But I wanted to try out what it would be like going into industry, and working on the education team here at Schrödinger has been really exciting because I'm still in that interface of I get to interact with scientists who are part of R&D, but I also still get to talk to students like you all today and introduce them to what a cool research area this is. So it's kind of the best of both worlds. So that's a long-winded answer, but hopefully that answered your question. Sarah, are the online certification courses free? So they do have a cost associated to them, but we also are aware that not all students are able to pay for these courses. So we offer free scholarships that you could apply for to get access to them. But you would be surprised, sometimes your teachers or professors have some funding available for kind of like these professional development tools. So it's always worth an ask, but we also have scholarships available if you want to apply for them, where you can get access to these tools for free. So again, shoot me an email and I can send you more information on how you could get access to that. From Rizabella, how did they get the drug-like atom number? So it's through a lot of different publications where they have estimates of how many different possible molecules are available, again, through machine learning tools and algorithms. So that's just an estimate and average from a variety of different publications that have looked into that type of research. Lots of questions, which is awesome. Any other questions?
Are the online certification courses free? That was one question.
Yep, so that one I answered. So they do have a student cost associated to them. There, I believe, are $125, but there are scholarships available for students who aren't able to afford that. Again, if you email me, I can send you information on how to submit a very, very, very quick application to get one of these scholarships. So again, feel free to reach out and I can help you with that. Any other questions? Great. If you think of any, again, please reach out to me via email, and we don't have to cut off the dialogue here. If you have any questions about career paths or how to get started in research, or how can you learn more about computational chemistry, please feel free to reach out. And I would be excited to chat with you all. Thank you so much.
So there are no further questions. Thank you very much, Katherine. And that was a very, very good talk in here. And it is very significant because it is contemporary and it uses a whole lot of the computation and all that. And we thank you sincerely for a very nice talk. And probably our audiences will be in touch with you beyond this particular seminar. Thank you very much, Katherine. And thank you all.
Thank you.
And thanks to
Thank you.
Daniel for arranging this seminar and plodding through that all the way. Thank you.
Thank you, Catherine and Krishna.
Thank you. Bye, everyone.